Welcome back to A&P. Today we are going over Chapter 22, the immune system. We've learned a lot about cells and tissues so far. And from what we've learned, you basically require a small army of all of these individual cells that have their specific function. We could be a red blood cell transporting oxygen, an osteoblast building bone, building cartilage. We could just be a regular cell breaking down and storing nutrients. What these cells have done is build and maintain a wonderful environment. It's moist, it's just the right temperature for the cells to be able to grow and flourish. It's surrounded by oxygen, by glucose. This is a pretty wonderful environment for a cell. And there are organisms that are going to try to take advantage of that. Organisms are trying to get into your body right now as we speak. They're trying to exploit that warm, comfy, nutrient-rich environment for their own growth. And those normal cells are basically unarmed. They don't have the ability to stop these organisms. That's why you have an immune system. A fighting force of specialized cells that defend your body against attack. They have a communication system all their own, they have strategies, and they have long memories of their past foes. They're your body's own SWAT team force. Let's give a quick definition of your immune system. Your immune system is composed of cells, proteins, and secreted products that all combat infectious agents. Infectious agents is just a broad umbrella term. It could be a virus, a fungi, a protozoan, a bacteria, or a parasite. But your immune system is there to provide immunity to all of these things. Here are some mugshots of those infectious agents. We have viruses, fungi, protozoans, bacteria, and multicellular parasites. Meet our leukocytes, commonly called the white blood cells. Immune cells are primarily housed in our lymphatic tissues, such as our lymph nodes. There is a specialized area for them to screen that fluid for any potential threats. But because our body is so big and each immune cell is so small, we have to just spread them out throughout the entire body. As such, we will find immune cells in select organs, like our brain, our skin and our mucosal membranes, and our connective tissues, which that can be our blood, our bone, our cartilage, our adipose, all over the body. Immune cell location is illustrated by this picture. We see them here circulating in the blood, located in our lymphatic tissues like our lymph nodes, our spleen, in select organs like our lungs or our brain. Our skin and mucosal membranes, we see here the dendritic cell, mast cells in our connective tissue. These immune cells are located throughout your entire body. But you know what we need? We need a way for one immune cell to communicate with the others. That way, if we have an attack, they can actually call for help and bring more immune cells to the site of the infection. So we're going to do that through cytokines. Cytokines are small proteins that are secreted by cells of the immune system. We're going to use these to do cell signaling, immune cell development, regulating inflammatory response, and destroying cells if need be. This table shows us some major categories of cytokines. The interleukin cytokine has the function of regulating immune cells. We're going to see this produced by our lymphocyte, T lymphocyte, macrophages, some endothelial cells, And if you ever see it notated, it's IL-1, IL-2. Another category is tumor necrosis factor. This cytokine destroys tumor cells. It may have other functions as well. We can't really ask a cell. We just have to try to see what effect a cytokine has on them. So it can be hard sometimes to find out what all it does. T lymphocytes produce these, macrophages, mast cells, dendritic cells, and it's normally notated by TNF, followed by a Greek letter like alpha, beta. A third category is the colony stimulating factor, or CSF. This one stimulates leukopoiesis in bone marrow to increase the synthesis of one specific type of leukocyte. For example, during a parasitic attack, will increase the amount of eosinophils we have. Interferon is the fourth one. There's three classes. These are either antiviral agents or pro-inflammatory agents, notated as IFN. These are four of the major categories of cytokines that your immune cells are going to use to combat an infection. Your immune system is divided into two types: innate immunity and adaptive immunity. When explaining immunity, I actually think it's time for a snack break. Steak and pasta with a little broccoli. Whenever you're born, how do you eat? Do you just reach with your hands and grab some of it? Eat that, reach with your hands, grab some more. Ooh, steak. You take it and you tear it. That behavior is your inborn way of eating. So that's going to be our innate immunity. You're born with it. You don't have to learn it. You just naturally do it. Then we adapt. We learn how to eat with a fork and a knife. This is called adaptive immunity. Now, adaptive immunity has to be learned. You don't know it automatically, like the innate grabbing with your hand. No one had to teach you that. If I were to take you out to a Chinese restaurant right now and hand you chopsticks, if you're not familiar with that, initially you're just going to be working on getting your hands right. And then you'll be practicing by trying to pick up a noodle. And then once you eventually get it, you're adapting. You're learning how to eat in a different way. The same way you had to learn as a child how to use your fork and your knife, now you're having to use chopsticks. But you can learn it, you can adapt to that. This is a good example of how your adaptive immunity continues to work for you as you get older. I'll finish my snack up here, and I'll see you guys back at the PowerPoint. Wherever you are watching this video, did you know that you are sitting there as a literal paradise to cells, to bacteria, to fungi? Inside of you, it's the ideal temperature for those cells to grow and divide it. You're covered in nutrients on the inside due to all that transportation of glucose. You have an aqueous environment. You're a good pH. You're protected from temperature extremes. Any cell that gets inside of you has it made. Because you are such a paradise, your cells have to work very hard to protect you. Your innate immunity is going to have one goal: prevent entry. It creates a literal wall around you. Probably don't think of it that way, but that's what your skin is. It's there to protect you and to keep bacteria that you may come across in your life out of your body. This wall is made up of both your skin and your mucous membranes. Because it is on your skin a very harsh environment for a bacteria, a lot of them won't survive there. If they get to your mucous membrane, we're going to have secretions and really sticky mucus that trap them, and then we can cough and sneeze them back out again and put them back on the outside. Unfortunately, life happens, and at some point, maybe you got a scratch, you fell down, that wall was torn. Now that you don't have the protective wall covering you, you're wide open. Those infectious agents now have free access to you. If you're like me, you've had many a scrape over the years, but I'm still alive, and so are you. That's because our innate immunity has a backup plan. Here's the barrier of skin over on the left. As you can see, all this bacteria is on his hand, but that's not going to harm you because it's on the outside of your body. But he was putting that block in his mouth. Now we have to use defenses like antimicrobial secretions in our saliva, in our lacrimal glands, in our nasal secretions. Also mucus and cilia in our airways to get something stuck and to push it back out. Hairs in our nasal cavity can help trap anything from going too far in. If it does make it down into our throat, we can cough or sneeze to remove it. If the baby does swallow the bacteria, it can go into the stomach where the high acid content is going to destroy it, or we can vomit to remove it from the body and get it back out. If an infectious agent does enter the body, it has to face your immune cells, your cellular defenses, your antimicrobial proteins, fever, and inflammation. These are all the weapons of the innate immunity. Immune cells can engulf unwanted substances. We saw that from our neutrophils and our macrophages. It can cause inflammation by releasing histamine and heparin. We saw that from our basophils. They can release cytotoxic chemicals to cause controlled cell death. This is called apoptosis. Your NK cells are really good at this. We can also release cytotoxic chemicals to destroy a parasite membrane. If we can put a hole in it, it's easy for the body to destroy it. Our eosinophils are good at this. Antimicrobial proteins include interferons. They're basically a flashing danger sign. If a cell gets infected, it can release interferons to signal all the nearby cells to, number one, synthesize enzymes to protect themselves, and also to signal those NK or natural killer cells and macrophages to come destroy the infected cells. Our big guns of innate immunity, fever and inflammation. For your average infection, this is going to be enough. But there are some cells, some viruses, some bacteria that are going to be able to overcome all of these. In fact, they might thrive in a fever environment. So whereas our regular cells are being stressed out by this high temperature, a bacteria that can flourish at a fever temperature is going to be doing just fine in your body and multiplying and growing in number. So we need to have a targeted SWAT team of an immunity. The body tried to keep the pathogens outside, but the barrier failed. Then it tried to use its immune cells, its inflammation, and its fever to destroy the invaders, but they failed too. The surviving pathogen is still at large in your body. Now it's time to use adaptive immunity, the specialized targeted defense system. Adaptive immunity is a delayed response to a specific antigen. going to utilize T lymphocytes and B lymphocytes. No longer are we using a system-wide inflammation response. Your entire body doesn't need that fever response. But instead, we're going to produce targeted cells. These are highly specialized immune cells that are going after a specific pathogen. What are these specialized cells? Your T lymphocytes and your B lymphocytes, now known as T cells and B cells. These immune cells are specifically trained in one of two ways. Your T cells, or your T lymphocytes, are immune cells that travel to find their target cell and destroy it. These SWAT team cells can actually spread out throughout the body pretty well. Like you're going to see specialized ones in your lungs or your brain for an immediate, but other than that, they might be going through the blood looking for something. This picture shows us what these guys actually look like. They're your round cell covered in receptors, shown here in blue and in green. Kind of like bloodhounds, they're going to be looking throughout the body. In contrast, B cells are going to be immune cells that produce antibodies. The antibodies disperse in the blood and bind to their target cell, marking it for destruction. Here's a picture of what a B cell looks like. These antibodies are the ones that work as the bloodhound, traveling through the body, searching for their pathogen. What are T cells and B cells hunting for? Antigens. An antigen is a substance that causes an immune response. Take the word antigen and you can break it down into causes antibodies to be generated. So antigen. An antigen can be a lot of things. It can be a chemical, a bacteria, a virus, pollen, markers on a blood cell, anything that causes an immune response. Adaptive immunity can be divided up into two categories. Either cell-mediated immunity, where a T cell travels to find the antigen, or humoral immunity, where a B cell is going to send antibodies out into the blood. We've already learned about antibodies before, but in relation to blood types. Your blood type was based off of what type of antigen your red blood cell carried, either an antigen A or B, both or none, and what antibodies you had in your blood. Anti-B antibodies or anti-A antibodies, neither or both. This is an example of humoral immunity because the blood is carrying the antibodies that were produced by a B cell. Recent estimates put the number of cells in your body at around 30 trillion. If you try to write that out, it's 30 and then 0, 0, 0, 0, 0, 0, 0, 0, 0, 0, 0, 0. With that in mind, how does a T cell find its target? How does it recognize that antigen and instead not focus in on a skin cell or a different cell somewhere else in the body just minding its own business? It does that using receptors. Lymphocytes have many receptors on their surface to help identify antigens. With there being 30 trillion cells out there, it's it's very important that these lymphocytes have a lock and key fit for their receptors, that only one antigen is going to set off and bind to a receptor. The cells activate after their receptor successfully binds to an antigen, but each receptor binds to only one specific antigen. Lymphocytes are covered in what is called receptors. A receptor acts a lot like a key. We don't have them as much as we used to. But when it comes to keys, each key has a slightly different shape. If a key is specialized enough, it should only be able to unlock a single lock. Two different lymphocyte cells might have entirely different keys on them. Remember, this is part of adaptive immunity. So these keys were learned. They weren't innate. They're learned due to past experiences, past infections. It's how we get immunity, is your body learns and knows these past keys, past receptors. Now that we've created a cell that's covered in keys, what are the locks? The locks that these receptors are looking for are cells. All the cells in your body are going to have markers on their surface. These markers can match up with one of the keys. If this happens, your lymphocyte cell is now activated. If it bumps up against the lock, it's not the right fit, doesn't unlock it, you just travel on, continue down the bloodstream. Then you bump this key into this lock and it's a match. Now they're activated. These locks are located on the outside of cells. Our lymphocyte, traveling through the body covered in keys, is going to be looking for the specific antigen that it unlocks. One of its keys is possibly a match for a lock on the cell surface. It just has to find the right one. A lymphocyte may have 100,000 different receptors or keys on its surface, each receptor only binding to one specific antigen. It's kind of like getting a big bag of keys, throwing a lock in there and shaking it up until finally one key lands in a lock and unlocks the lock. It's kind of hard to find a match. If your particular key does fit into a lock, that cell will be activated. You now no longer have a lymphocyte just traveling through the body. Now it is an activated lymphocyte and it's going to be able to attack and help us overcome whatever antigen set it off. A B cell has receptors on its surface called B cell receptors, or BCR. BCRs are going to have the ability to bind directly to an antigen. Once they do, it activates the B cell. So initially your B cell is going to be in relaxation mode. It's looking, but it's not activated. So it's plain clothes. There's your B cell. Our B cell is covered in those BCRs, the B cell receptors, and they're looking for an antigen. Suddenly, one appears. That's the lock and key fit. And what is this lock on? It's on a large cell. Now, the B cell is activated. Before it was relaxed, and now it's angry. Sorry. All right, so we have an activated B cell at this point. When that occurs, whatever receptor was able to bind to that lock, we are going to start production. We're going to create more of those, and more, and more, and we're going to send these off through the blood, thus humoral immunity. And all of these antibodies, they all are looking for the lock that this cell carries. So once activated, after that initial contact, your blood can become so saturated with these antibodies that every one of these cells is located and found in the body. So if you had an infection and you're being overrun by these cells and they were able to escape the innate immunity, maybe they can't escape the antibodies because they are going throughout the entire system hunting down that antigen. So the B cell is going to create more memory B cells. These B cells are covered in the same receptors that were activated by the initial B cell. That way, the body's going to retain a memory across the cell generations of the receptor that was activated by that antigen. Since T cells are able to create such an immune response, causing cell death, we have to be very careful about how they're activated. We don't want them going off for any antigen. The body thus has a second layer of security. The receptors on the surface of the T cell require two things to activate them, not one. They have a co-receptor protein and their T cell receptor, or their key. These are paired up over the body. Your particular key or receptor and a co-receptor protein. Our B cell was able to have its BCR, B cell receptor, bind directly to the antigen on the cell. This then activated the B cell and it produced the antibodies. In comparison, our T cell is going to need to have the receptor, the TCR, be a perfect lock and key fit, and the co-receptor protein has to bind as well. Here's a simple antigen. It's located on a cell. The BCR could bind to this, but your T cell, it may be the correct key, but because it's only the antigen and there's nothing for the co-receptor protein to bind to, this will not bind. That's just to protect the body so we're not constantly activating our T cells. Instead, a very interesting process occurs. Large immune cells are going to come along, have a lock and key fit, and engulf that entire cell. Phagocytize it. So we're going to see this from a macrophage, from a dendritic cell. They engulfed the entire thing. But they didn't just destroy it. They took that little lock and or antigen that was on the surface of the cell. They took it, kept it, and presented it on their surface. And in addition, they held it out on a structure called MHC. Here's another MHC, specialized structure, holding a copy of that antigen. When an immune cell engulfs and phagocytizes a pathogen, which it could be a bacteria, is a good example of one, they can take that pathogen and break it down, but retain the antigens. Take the antigen and present it on their surface, being held by an MHC or Major Histocompatibility Complex. By presenting the antigen on its surface, it's asking to be bound to by a T cell. When the antigen is presented on the surface of the cell using the MHC, it allows the T cell to have both the TCR and the co-receptor bind. When this happens, now the T cell is activated. Just the antigen alone on that bacteria's surface couldn't activate the T cell. It needed to have it being held by the MHC. With T cells, we actually have two types, a helper T cell and a cytotoxic T cell. A helper T cell is your immune system commander. This T cell has what's called a CD4 protein, and when it's activated, it releases cytokines. Cytokines are how these immune cells communicate. It can call other immune cells and help command them in defense of the body. Cytotoxic T cells, in comparison, are going to release toxic chemicals when they're activated. These have the CD8 protein. When activated, they destroy infected cells. How about a roadmap of all the ways adaptive immunity can help protect you? A virus has entered the body. The innate immunity was not enough to stop it. It made it through the skin barrier into the body. Inflammation and fever weren't enough. As the virus travels through the body, it's going to be infecting cells. When that happens, these infected cells can try to present the virus antigen on their surface as a way to alert the body. A cytotoxic T cell was able to bind to the MHC complex holding the antigen. When it did that, it released the toxic chemicals and destroyed the infected cell, thus stopping it from replicating those viruses. It also produced some memory T cells. That way, even after this T cell passes on, other T cells can remember it and produce more of those receptors. A different virus came across a macrophage. The macrophage engulfed the virus and phagocytized it and presented the antigen on its surface using the MHC complexes. A helper T cell was able to bind to those. Upon activation, it produced cytokines to communicate with the cytotoxic T cells and the B cells. It also produced memory helper T cells. The helper T cell, remember, is our commander. So it uses the cytokines to tell the cytotoxic T cells where to go and what to attack. It also tells the B cells, activates them, gets them ready to produce more antibodies. This virus was able to bind directly to the B cell receptor. When it did, the B cell began to create plasma cells to produce antibodies and also memory B cells. So it may have only been 24, 48 hours since this virus first came into the body. But so far, it's being destroyed by toxic chemicals from the cytotoxic T cells. The helper T cells are aware of it and helping to command the immune response. And your B cells are pumping out antibodies that are traveling through the blood looking for that virus. This is how your body protects you. The antibodies that your B cells are producing are tags. They're created to mark a specific antigen for destruction. Another name for them is immunoglobulin proteins, or Ig proteins. An example that we've learned about before is that an anti-B antibody will bind to and tag the B antigen. Now, your antibody is divided up into two regions. The base of it, or the stem, is called the Fc region. And it's this portion that's going to interact with the immune cell surface. The variable region is up here at the arms of the antibody. The variable region has on its end the antigen binding site. It's called variable because each one is specifically designed for a different antigen. If you were to pluck at random two antibodies from out of your blood, their variable regions would be completely different, each one being designed to bind to a specific antigen. Here's one binding in the variable region. Actions that the antibody can take using the variable region include neutralization, agglutination, and precipitation. Neutralization is when the antibody, with its variable region, binds to every available binding spot on the virus. When it does so, it covers up any other spots that might bind with the cell and prevents the virus from interacting with the cells around it. Agglutination is another tactic they can do, where the variable regions bind to two different antigens. So two different bacteria can be bound to by one antigen, creating a clump. This clump stops the bacteria from accessing other parts of the body and makes it easier for it to be removed. Precipitation is when the antibody binds to toxins and creates a complex that is insoluble and can be moved out of the blood. The Fc, or the stem region of the antibody, has actions as well. These include complement fixation, opsonization, and activation of NK cells. With complement fixation, an antibody that has bound to an antigen on the variable region will look for a complement protein to bind to its Fc region. Opsonization is where an activated bound antibody is looking to bind to any phagocytic cell with its Fc region. When it does, it triggers that bacteria to be phagocytized or destroyed. Major classes of immunoglobulins, or antibodies, include IgG, IgM, IgA, IgD, and IgE. Each of these have a primary location where you find them, as well as specific actions they are able to do. They also have unique characteristics and different lifespans. For the lecture exam, this is the summary information that you have to know on 3 of those antibodies. Allergies are any abnormal immune response to an antigen. It's considered an allergy when your body is responding to a non-infectious substance. Instead of an antigen on a bacteria, it's an allergen from a plant. The response is going to involve your IgE and your pro-inflammatory cells. If the body goes into inflammation, it can lead to hives, asthma, or anaphylactic shock. It's considered an allergy if the response occurs within seconds, and a hypersensitivity if the response takes hours or even days to occur. It's likely that whatever your immune system comes up against, it might see it again. Because of this, we want to have memory cells, cells that are going to retain the receptors for specific antigens. The first time your body is exposed to an antigen, it slowly produces antibodies and memory cells. It takes it a while when it meets an antigen for the first time to create those. This is called your primary response. So the second time you meet an antigen, after you've already had a primary response and created all these memory cells, the second time it's almost instantaneous. The secondary response is very quick. This chart shows us the primary and the secondary response. It measures the amount of antibodies that are being produced. On your primary response, it took you maybe 12 days after you first were exposed to an antigen to reach peak antibodies for IgM, and it took maybe 17 days to reach peak for IgG. And you didn't produce all that much antibodies. But you produced a lot of memory cells. So years later, when you're exposed to that same antigen, that secondary response by day 5, your body is covered. Your blood is just saturated with antibodies. IgG is at maximum production about 5 days after you're first exposed to it. You can imagine the immune response here with those antibodies circulating in the blood, tagging that antigen, doesn't have a chance. The primary and secondary response is how vaccines work. To vaccinate someone is to administer a weakened microorganism or its products, just pieces of that antigen, to produce an immune system with initial exposure. It's going to give you that primary response that's pretty slow, but allows you to develop those memory B cells. That way, if in the future the actual pathogen that had that antigen invades your body, your immune system's memory cells are right there and create an instantaneous, powerful secondary response, thus destroying that infection before it even has a chance. Unfortunately, those memory B cells don't live forever, and you may have to give booster shots over time to remind and get that primary response going again, make more memory B cells. There are 2 ways to obtain immunity. One of them is active immunity. If I'm exposed to a virus just going about my regular day, that is a natural way to acquire active immunity because that virus is covered with antigens that my immune system is going to be exposed to produce memory cells. If I go get a vaccine, that is an artificially done active immunity because I artificially exposed my body to those antigens and it produced memory cells. Passive immunity is the acquiring of antibodies from someone else, an individual or an organism. We naturally do this as a baby drinking breast milk, as the breast milk contains antibodies from your mother. Or we artificially can separate the antibodies out from someone's blood who's already fought off that infection and give it to someone who's currently fighting it. Whew, that was a lot of information. The immune system is a pretty big chapter, but it's an important one. And with that, we wrap up Unit 2.